thank you for organizing. It's been a pleasure to be here. So yeah, today I would like to talk about three projects that go sort of through my career trajectory. The first is Ethoscope. It's going to be about circadian rhythm and sleep in insect and how to monitor that building hardware and software. The second is Sticky Pie. It's a smart insect trap I built in my postdoc. And the last one, it's a project that will start next year. It's called Global Warming. I'll let you imagine what it's about. Surprise. So the Ethoscope, first of all. So in my master's, I studied, so I'm trained in biology, right? And I started being interested in computer vision and image processing, and I kind of wanted to do that and use it as a biologist. And I met this chap on the right, Giorgio, with the sunglasses, and he was starting his lab in Imperial College London. He works on sleep and fruit fly, but he wanted to move on and change the way we study sleep. So I took part in building this team, and really it's about circadian rhythm. So as you probably know, and it's been in the news a few years back, because it was the Nobel Prize 2017. Most organisms on Earth, plant, insect, mammals, birds, have a circadian clock. And it's an internal clock that ticks, and this is why you experience jet lag when you move to another time zone, right? And the clock is involved in many physiological processes, like reproduction, immunity, development, feeding. All these things are timed, and it's extremely important ecologically, because organisms live in this 24-hour periodic environment, and if you need to do well, you actually have to predict the future a little bit, and you can only do that if you have an internal clock. One thing that is really attached to the clock and that we experience hopefully every day and night is sleep. For a while, for almost 20 years now, sleep has been studied actually in Drosophila, in fruit flies. And the reason we think fruit flies sleep is because if we put here on so imagine the y-axis for now is the probability of being immobile of an animal. So around dawn and dusk. So here we have the start of the day, the white is the day, the dark is the night, and we have a low probability of being immobile around dawn and dusk. But then during the day and during the night, animals are quite immobile. And what happens is if you prevent them if you don't allow them to be immobile by shaking them, for instance, then the next day they need more immobility than they would. And this is something we experience as well. If you don't sleep one night, you will sleep more the next night. This is what we call the sleep rebound. And this is something that in mammals is involved in learning and memory, but the function of sleep in general and whether it's the same thing between invertebrates and vertebrates is actually very unclear still to this day. Now, if we think of hardware, how do you do that in fruit flies? Well, most of the community has been using this very sort of low-tech principles, and that's been used for dozens of years. Where you have an infrared beam that crosses a tube, you host the fly in the tube, and it has food on one side, and you can keep a fruit fly there for more than a week, and then you just can replace the tube, and you can keep them for their entire life. So it transforms a complicated problem about behavior in a one-dimensional space, to whether they move in the tube. As you can see, this is great to study walking. They will cross this midline when they walk. But if they move a little bit, and we know that fruit flies grow, fruit flies feed, fruit flies females lay eggs, all of that you will miss because they're not actually crossing the beam. So you capture walking activity, and this is a great tool for that. But as you want to study something like sleep, you want to account for these other, what we call micro behaviors. And this is where we decided to use a camera-based approach. So we built a setup based on the Raspberry Pi and the available camera at the time. This is an infrared camera, and then we bounce infrared light back up, and we can monitor fruit flies also in tubes, 20 tubes at a time, in a 3D printed setup for multiple days. This is a cost-efficient setup. It was about £100 or so per device, and by the end of my PhD we had something like 70, 80 devices in the lab. So the type of thing we see with this camera is not very high resolution, but we can see this is a fly walking. This is a fly that is by the food doing something, that it could be feeding it, but it wouldn't cross a beam, right? And it could do that for a long time. And this is a fly that is, as far as we can tell, completely still. The nice thing with this setup is that it was very customizable. So we could film the insect, but we could build module to do new things on top of that. One thing we quickly realized is that this generates a lot of data because of this 80 devices, 80 fly per devices, and we operated around 2 frames per second. So we actually built a collection of R packages to process this data and also to process the legacy Drosophila activity monitor data. So there were a lot of issues regarding labeling and computing circadian statistics. But essentially, in the end, what we were able to do is move from a paradigm where we had walking versus not walking to something that was a little more subtle with video that was walking completely still and doing these things that we couldn't really score too well that we called micro movements. So in the end, we could, I'm trying to play this GIF, but let's see if it works. Otherwise I'll come back. It's okay, I think I will skip it. Essentially, each point here is a fruit fly, and at any point in time, it moves in this behavioral space, and we can then not only study where it is in this space, but how it transitions from one state to the other. But where it gets interesting is that we can actually start sleep depriving fruit flies in a much more interesting manner that we were able to. So the reference paper when I started my PhD was a 2002 paper that said fruit flies die when they are sleep deprived. Now this is crucial because if, as a biologist, when you want to study the function of something, you remove it. You want to study the function of sleep, you try to remove it, you see what happens. The literature, which was all going down to this paper, said, well, we took a few flies, that was 12 flies, and we sleep deprived them. Now, this was by looking at the tubes, and when the fly stopped, you shake them to wake them up. So that must have been quite a few sad students doing that for 70 hours, right? Which explains the low numbers, but it's very anecdotal, although crucial. So 2 flies died after 60 hours, another 2 died after, and then this is why sleep is a vital process. Yeah? So this was the state. And we thought you can basically program this machine to do that. So the way you do it is you analyze behavior in real time, and essentially what you can do is you have the immobility of the fly, and whenever it stops for more than a certain set duration, then you prevent this from happening. So you can essentially sleep deprive them. So this blue line where we rotate the tube when they stop, they don't experience much immobility, and then in the morning, as expected, they rebound. Yeah? Now, if we can also show that if you startle them as they just walk and cross the midline, they don't rebound. So we can do this type of control. And what we were able to show is that actually when you sleep deprive flies for much of their life, they do not die, and it's not as dramatic as you'd expect from the literature. So it was a little bit of a negative result, but we couldn't replicate lethality from sleep deprivation. So I don't know if sleep exists in fruit flies, to be honest. But what we also were able to do is do other kinds of sleep deprivation. So it turns out that when you put females with males, they are sleep deprived. So you can put a female all night and the males basically do not sleep. And that works if you put pheromones of females as well. But the funny thing is they don't need a rebound after that. They don't rebound. So it's not the same thing startling them or providing them with a sexual stimulus, for instance. The one last thing that my friend Alice French did not too long ago is that actually flies, so we adapted the system to also be able to send odor in the tube in a programmable manner. So if a fly stops for long enough, you can send a puff of odor. And she showed that indeed you can wake flies up with odor, and it's not really the concentration of odor, but it's really the biological meaning of odor. So it's more the salience of a stimulus than the actual intensity. So after kind of torturing flies in the lab, forcing them to stay awake, I wanted to move on and do something more applied to ecology. This is where the Sticky Pie project came about. I was in BC, in Canada, and I made this man on the right pole, and he said, "Quentin, I read your papers on sleep. I'm interested in sleep. I love parasitoid wasps, and I care about the ecology of sleep, and I think they sleep at different places." We had a discussion, and I thought, but how do you monitor activity of insects, like circadian rhythm, in the field? This is quite different from the lab. You can't put them in tubes, or it's irrelevant. So how do you do that? And we were discussing, well, in the field often you use insect traps to capture insects. What if you could have a timestamp every time you capture an insect? And this is where we started going. So we had this crazy idea of making a trap that has a little door, and the door opens at different time of the day. And in the end, we sort of started reading the literature, and there were so many traps around. There were smart traps, but a lot of these at the time were designed for agricultural purposes to kind of make human work higher throughput. So they tell you the same thing as a regular trap would, but they didn't have the temporal resolution we wanted. We wanted something that could tell you pretty much, 3 times an hour which new insects came about. So we wanted the frequency, and that was something that we wanted for research, not for pest control. And really the hypothesis here is that if you study different groups of insects, they are differently active at different times of day. You would trap them at different times of the day, but also things like the weather might be factored in, and you could predict, you could essentially have a forecasting system for insects present. So in the end, after all this discussion, we decided to kind of make the problem simple, and it's an approach that others have taken in the literature, is that we put a camera in front of a rectangular surface, which is a yellow sticky card. I bet a lot of you have seen these. They are sticky, insects fly around, they get stuck, and they stay, presumably for as long as your experiment lasts. So this was really something that could stay autonomously for 2 weeks in the field, take an image every 20 minutes, and we care about, of course, seeing insects being trapped in the night. So we had a flashlight in the back, so we can illuminate through and have sharp, contrasted images. This is for an example of a day image. So this is the full resolution, and then zoomed in. So this is one of the wasps we're interested about. It's about 3 millimeters long, and at this resolution we can start identifying insects that are about 3 millimeters if you know what you're looking for and they are distinctive. But more than just one trap, we really wanted to build a platform where if you deploy multiple traps, you can gather the data together. So now in Vancouver we have about 50 traps that are deployed, and you can retrieve the data and put it on a server. I'll just show you if it works. So this is the type of web interface we built. You log in, and then you can select the time window you're interested in. So for instance, here you have 22,000 images, and then it displays variables you might be interested in. So each trap has a temperature and humidity sensor. So here it shows in blue humidity and red temperature, and you have a thumbnail of the image you're looking at, and you can click on images, and it shows you the segmentation, which is the first step of the processing. So you can do some quality control. So it's all open source, and there is a documentation. We explain how to build it, how to use it. And this is the type of image or videos we get if we look at it. So we kind of thought insect would be stuck and that's it, but it was a lot messier than we thought, actually. And it's not something we knew. It's something we thought you put a trap there, insect gets stuck, you retrieve everything that was stuck, but we had all sorts of things happen. So insects, first of all, don't get stuck near as much as you think. Some insects escape. You see the slugs around? Sometimes slugs come and eat your insects, which is really interesting. And then, of course, you have artifacts, fog, and some insects are really good at escaping. You see them in just a couple of frames. So it became more of a messy problem than we anticipated. So we ended up making an image analysis pipeline. So throughout my PhD, it was very simple background abstraction, and I was really craving for, as my PhD moved forward, all these fancy techniques started being made in deep learning, and I wanted to know what it was. So I thought this is a great opportunity. But I wanted to break the problem into something tractable. So we have a series of images through times, right? That are about every 20 minutes. And I reasoned, let's first not care about which insect it is, and we do an instance segmentation, which I call the universal, it's a little bit pretentious, I realize that now, but a universal insect detector. We do instance segmentation and find all insects indiscriminately. This is important because you've seen there are sometimes partial insects, or there. So what for me, it doesn't. I don't want partial insects. I just want when the insects are complete. And then I wanted to track them. I wanted to track them because I don't know if you've seen, for instance, sometimes it's foggy, it's rainy. If you do not track insects and you just see what you have and ID them on the spot, if it's foggy for 12 hours and then it's not foggy anymore, you will discover lots of new insects that all of a sudden everyone is there in the morning. So I wanted to see, okay, this insect was already there. So tracking was important. And then we end up with these tuboids, which are kind of tracklets that are a series of arbitrary number of shots of the same insect that, as we've seen, they can move or they can be occluded. So, for instance, here we have this beetle at multiple times, and we want to reconstruct this trajectory, but also then take every tracklet, every tuboid, and make a single prediction for each tuboid. So the first step was the universal insect detector. For that, we made a reference of 500 images. At first, we just used images we had, and that worked okay. But we noticed that actually including similar images from literature, like moth traps and so on, improved prediction on our own image. Quite surprisingly, I guess it's not surprising for most of you, but at the time for us it was, that you can abstract more what is an insect, and it works better in your data if you include foreign data. So we used mask RCNN. We've heard about that a lot this week. But you can see that the problem, as you see here, so you really have, so in blue, those are kind of false positive. So here is an image that we detected, and this is the ground truth, right? And we have false positives or false negative, but of course when an insect is partial, it's a difficult problem. So you're always going to miss something that is at the head of an insect. You're not completely wrong if you say that it's an insect. But all in all, we reach really satisfactory in distribution results. And what we noticed is that here is the recall, and we have the area of the insect that we're predicting. So we are much more sensitive beyond the sort of 1,000 pixel surface insect. And prior to that, we actually miss a lot of insects, which is not surprising. But what's good is that the precision was pretty constant throughout. Then for tracking, this is something that can be, I think, done much better these days with a lot of new tracking tools. But what we thought is we looked at frame and reasoned how do we know that it's 20 minutes apart, right? So we don't have a very high temporal consistency for those insects that are actually capable of moving on the trap. So how do we know an insect is the same insect between two frames? So instead of thinking of it as the entire trajectory, if we compare pairs of frames, perhaps we could reconstruct the trajectory of an insect. So if we have an insect here on the frame and a candidate insect on the next frame, how do I know it's the same? So for that, we built something based on SAMIs or triplet type of architecture, where we have a network with the same weight, and we feed it with the candidate insect, with the real reference insect, and then we kind of applied the mask of the first insect on the second insect. So that's to capture that. One information we use when you do this is we don't just look at two images, but we look at whether the insect in the second image is missing from the same location in the first image. But instead of just using visual similarity, we build a network where we add also the ratio of area and the actual displacement of the insect. So this is probably actually a lot more informative than just the similarity. But we then build a sort of explicit and implicit mixture. And then we sort of greedily apply between all pairs of frame an all versus all match, and we essentially construct a directed graph through time that we can then patch to account for long range matches after, say, 6 hours of fog or something like that. So in the end, it gives us something like that, which is sort of difficult by hand to evaluate when it goes wrong. But this is the sort of thing that you can't, your model will tell you I'm 99.99999% accurate, but it doesn't really matter because the errors propagate, right? So if you have 100 insects and you're wrong once, this is quite a lot. Okay, so that's that. And then we moved on, given this cuboid, this stack of multiple insects, how do you predict what it is? And the sort of default would be I take the first frame and I feed it into a ResNet and I predict one of the class. We handcrafted 18 taxa that we sort of entomologically made sense in this field site, but instead of just using the first frame, we thought this is kind of a waste to not use the other frames in the same stack. And that's because often you can think about it as a kind of physical augmentation. You have insects that are spotted in the day, in the night, they move a little bit, so it seems unreasonable to just use the first frame. Then sometimes the first frame is wrong. If you've not stitched your images well enough, you have outliers where you have a ladybug that transforms into a weevil, for instance. So what we did is we take the first frame because we think the quality of the insect is better. Sometimes insects degrade over time, they get eaten and such. So we take the first frame and then we sample another 5 frames from the rest of the tuboid, and we compute a feature vector with a ResNet, and then we compute a median along this feature vector. And this is what we actually use for making the prediction. And we pick medians to get rid of these outliers, so that if you have a really unambiguous ladybug and the rest is actually a weevil, we don't actually move our space towards the ladybug. Does it make sense so far? So then we, as I said, we handcrafted different classes, and this is where I'm not completely satisfied with how you sometimes use ResNet for this type of problems, because we had a class that is this is background that somehow we still detected, and then we had something that we know it's an insect like this one, but I have no clue what it is. I know it's an insect. It looks like an insect, but this is it. And then we had other classes like other Coleoptera. That is, this is a Coleoptera, but I don't know what it is in this dataset. But within that, we have those click beetles. That we know what they are. So these classes are ontologically nested, but we force them to be discrete. But this is what we did, and it kind of worked for what we wanted. What we cared a lot about in this work was male spotted ring Drosophila, that we ended up having a decent precision and recall for, and they were quite abundant, so we thought that would give us a good idea of the trend, the ecological trend. So we went on and analyzed the results of our experiment, where we wanted to check that this type of tool could be used to monitor circadian activity. So spotted ring Drosophila are attracted by things like vinegar, apple cider vinegar in particular, and we had 10 devices. Half of them were baited with apple cider vinegar, and the other not. And we cared about both the male spotted ring Drosophila and also also parasitoid wasps that are, in this case, a lot of them that we found in the field, they lay their eggs inside the larvae of spotted ring Drosophila. So now if we monitor, this is the rate of capture per trap per hour over the season, we're actually able to reconstruct plausible data where on apple cider vinegar traps we have these spikes, and we actually could statistically show that this is because the bait diffuses much quicker than we thought, which is really interesting. That if you do insect trapping with baits, the kinetic matters a lot. It's not something you could recover if you have a static trap that stays there for 2 weeks. So you could say, I trap my insects with ACV, and even a day after, your trap is much less efficient. And unsurprisingly, this is true for male Drosophila suzukia, so spotted ring Drosophila, and this class, other Drosophilid, it was mostly female Drosophila. The nice thing with these sticky cards is that you can recover them afterwards, and we validated a lot of these classes with DNA barcoding. So you have the samples, and if you have the time and you can spend the effort, you can pick one and match it to the image in the video. Yeah.
Like you can match it locally, like that specific individual.
Yeah, but as you see, they move, and it might have disappeared and be eaten or something. So it's not always possible, but it can be if it's especially for late-ish insects. Now, if we wrap time and look within a day, so this is a weird daytime where we essentially warp time so that, because of course the day length changes along the season, so we just warped it so that 0 is always the start of the day, then 12 is always the end of the day. And then, as expected in the literature, and it's also something we've seen early on in fruit flies in the lab, we have animals that are active so in between day and night. So we call them crepuscular, and it's something that before, to capture that, you needed a lot of effort. So you needed people who camp in the field and retrieve those traps hourly. But hourly is not really sufficient if you want to study subtle variations in something as strong as circadian rhythm. So if you want to see a change, often the change will be 20 minutes. Like if you are earlier to go on a resource 20 minutes than you would otherwise, that's a lot. Those things are pretty consistent. So for our fruit flies, they were active at dawn and dusk, but you can see that the wasps, the parasitoid wasp, it's actually diurnal. It's an extremely diurnal insect. We didn't really yet analyze the effect of weather. It's something that would be really interesting in terms of predictive power, but you see here, it looked like an artifact. But if you actually look at the weather data and the images, this is a really rainy day. So you can start accounting for these things, and really, it's like moving from climate to weather in terms of resolution. If you want to predict weather well, you need hourly weather data. If you want to predict insect activity well, you need hourly insect data. And we wanted to generalize that to the concept of temporal niche specialization. So we have talked a lot about these ecological niches. That could be your host plant, that could be anything, but you could also argue that the time of the day you're active at is a niche. And we wanted to see how within a community these niches are partitioned. So we did another experiment, still with the same 10 devices, but this time no bait. Just so to go over this very descriptive part, we have, those are the mosquitoes, and as expected, they are active mostly at night. And so were the very few moths we could identify. But then we have expectedly diurnal insects. So hoverflies, it's hard to think of an insect that would be more diurnal than an hoverfly with a giant eyes. And same for the wild bees we had here, and the sort of housefly looking insects. But then there were things, so those five were things we kind of knew from observation. But how do you know that? Like if you read a book and someone tells you, well, this insect is mostly active at night, this is not sufficient for me. I want to know the numbers, I want to know a distribution. And for those insects that we didn't know well, this was really interesting, where we could see, for instance, this leaf hopper had this very interesting sort of evening behavior, and that's just not something that is easy to document. But that also means that if you go out and observe insects or catch them, you need to think about what time of the day you do that. And we're able to sort of formalize that a little bit more by embedding the distance between each population of insects and sort of see how these communities partition. So altogether, that means we can go beyond the anecdote of seeing this insect at night, but put numbers on that. Yes, so meanwhile, this is an ongoing project. The device has improved, and in a few ways that I would be happy to discuss. One of the main improvements is that we have a phone app to retrieve the data, and the data retrieval is much smoother, so we can come up with a phone, retrieve data from multiple traps at the same time, and the phone app sends this data to our API automatically. There are different alleys. The thing I was not really satisfied with, the classification of classes that are nested within each other. Now Melika, who's here and whose poster you must have seen, if you haven't, it's probably still here. Melika is working on hierarchical classification. Also, we're investigating using metric learning and this sort of things. So don't hesitate to talk to us if you have suggestions, ideas, or whatever. Daphne, who's in Canada and visiting Europe soon, she's working on the effect of artificial light at night on insects. So this is really a problem that requires monitoring a long time. So she builds her own hardware to impose a treatment and uses sticky trap and other trapping system to monitor insects. And then I wanted to introduce also some of the, so now I'm in Aarhus, right? And a lot of us in Aarhus, and this is just a few of the people we have, but we're sort of consolidating a computational entomology team. So don't hesitate to stop by and talk to us. We have a Talker High's team. We have also Kim Biak, who's been developing some of the tools Luca has presented already. One of the tools they have developed is this thing called BioDiscover, where you take an insect, a large sample of insects, and it sorts the insects and takes images of them as they get through a tube. And they're developing a more robotic system to kind of be a digital entomologist, essentially. So this is really exciting for us to work together. We have the moth trap that Luca has presented that Kim developed, which is very similar in idea to the sticky trap, except that it needs to illuminate at night constantly and it doesn't stick. And Kim has also been working on hierarchical clustering, so if you're interested, I invite you to read this recent paper. And the last thing that I wanted to mention is we also study insects in the context of food and feed, with Sarah's work on black soldier flies, and sort of using phenomics, using images to predict weight or sex of insects in the context of breeding. As Luca mentioned, this community of computational entomology is growing, and we are part of this cost action. So please stay tuned and look it up, Insect AI. This is led by Tom August. So we hope to see more of you there if you're interested in insect monitoring using AI. And that leads us to global warming. So
It's like a great time.
we've heard a lot about insects and birds and trees and like plankton and stuff, but where are the earthworms? So when you think about it, actually, more seriously, if you take a square meter of soil in a temperate region, you find dozens and dozens of earthworms there. The biomass is considerable. And soil in of itself is an extremely important resource that we're losing. So we need to think about soil more and about the organisms that engineer the soil. We've seen some of the organisms, we've seen the microorganisms, but there are microorganisms that we've left alone for way too long. And this is where I sort of wanted to start a project on earthworms. Hello. Yeah, no, if you actually look at the literature and what methodology is used in earthworm science, it's a lot of it, except for the eDNA, is actually something you could have used in the 19th century. So you dig, you count, you sort, and that led me to be fortunate enough to obtain a 5-year funded project where we can hire 3 people. So there'll be 2 PhD students and 1 postdoc working on these 3 packages that are really about using machine learning, AI, building hardware to monitor earthworms and especially novel aspects of their biology. I've just shown how you can use AI to recognize insects, and dozens of teams are doing that now. But earthworms are kind of difficult to image. They're sort of squishy. They live in an opaque environment. Like how do you
scanner?
Flatbed scanner. That's a good idea, but how do you, like, you need to see the anyway. There are lots of problems with that. So you need a way to consistently scan them, even if you want to do that on museum collections. You need standards. These are images from GBIF, and if you show that to a taxonomist, they will laugh at you. This is not sufficient to actually ID an earthworm. It is just not. So it would be pretentious to say we can train. Your algorithm will learn the context. It can't do anything with that. We're talking to people who, looking at the reproductive organs of earthworm on a microscope and counting the segments in between, right? So the idea here is really to build a system that I call the Veriscope. That's a prototype that does not exist, but in this case it's a scanner that just goes around an earthworm and renders a tubular image, right? And then we could use another an algorithm that for now we call deep worming that also does not exist. No, they exist. It's not started. So the idea is really to think, well, this earthworms are segmented, right? They're made of extremely visible segments, so this is a segmentation problem. So if we could kind of parse those segments and really kind of extract taxonomic and morphometric information, use concepts from actual natural language processing on this sort of thing, right? So to understand the grammar of segments, if it were, and is this grammar something that helps you to predict the species? So it's a little late, but there is still time. We're recruiting a PhD student to work on this very first work package. 7 days left. But there is another work package which is more about the ecology of earthworms, how earthworms interact with plants. Now, for insects, we know all sorts of things. Insects can be herbivore, they can do pollination, they can do seed dispersion. You have tritrophic interactions where a plant attacked by an herbivore can recruit a predator of these insects. So super interesting stuff. But for earthworms, we've just assumed that these things that have lived, so plant roots and earthworms that have lived together for millions of years are passive to one another. I don't think it's as simple. I believe there are interactions between them. It could be tritrophic. It could be that when a plant is attacked by a fungus, it can recruit an earthworm to eat the necrotrophic fungus or this sort of thing. But we need a system to look at that. What the colleagues do to address this type of question, they put plants in 2D terraria like this one, and you can see the earthworm, you can see the plant, but they sort of put them there, and by the end of the experiment, they take a Sharpie and they draw. So you don't really get behavioral resolution here. So if you just put a camera instead of this person, if you replace this extremely talented researcher by a camera, then perhaps we get slightly more information in addition to the expertise of the person. So the idea is really to make that higher throughput by co-opting a conveyor-based system in greenhouses and having basically those setups move in front of cameras. Again, this is not started. This starts next year. So hopefully the student who does that finds a much better way to do it. And the last thing that is the postdoc project, so that's 4 years of postdoc, is to actually move on to remote sensing earthworms. And that's just an example of a review in 2020 that lists all the insect traps that they could think about. And there are also large industry that makes insect traps. And this is something that is really popular. But how do you monitor earthworms with the same type of technology or equivalent ambition? So as we said, soil is opaque, soil is 3D, like it's very heterogeneous. It could be sandy, clay, wet. So the solution here is to improve what already exists. So this low-hanging fruit is often you go around, and if you walk on a lawn like this one, you'll see, actually, I hope you start noticing more if you haven't yet, you'll see all of these casts. And these are earthworms that we know go out at night and forage litter. And if you just put a camera there, you can actually see them going out probably, and you can see the casts being made. So that would be an idea of their behavior. At least this type of earthworms, which are sometimes a minority of the population. For earthworms that don't go back up, often people put those baits here. So this is called the laminar bait trap. You put baits and then you remove it, and then you count how many baits have been consumed. I think we can do that with simple sensors. Essentially replace this by a PCB and have simple sensors and little holes to monitor both the soil physics and the biology.
Ivan, can you close?
Thanks. Yeah, speaking of noise, we can also probably use acoustics. This is something that has been actually experimented in the lab, and it can work in the lab to listen to earthworms moving in the soil and to roots growing. But I think it may be extremely hard because the real world is noisy. There is wind, there are root growing, there are cars, There is ambient seismic noise. So this part, we'll see if it works. But it'd be cool to be able to listen to the earth tone. And if we do it, it'll be quite, it'll have to require a lot of processing and probably things like arrays of microphones or something like that. So yeah, if you're interested in this earth tone project, I'm really happy to discuss because it's not even started. In summary of today's talk, it's really kind of a spectrum of how building new tools for ecology is not actually so hard, and you can build tools that encourage you to ask new questions. You can use tools that increase your throughput, but you can also use tools that allow you to do something you would otherwise not be allowed to do, so provide new content. And yeah, I think altogether we need a community of ecologists that are more involved in making, like maker ecologists. And on that note, I'd like to thank you all for listening. Thank you for inviting me. And that was work in the Gillester lab, in the Haney and Carilla labs, and now in my lab, Darsa. So thanks.